It's time for another session, and I'm really excited about this one as well. I believe that this session will collectively open our eyes to the potential that more study of the retina could have on the way we develop Sanfilippo treatments and assess their efficacy. So please welcome our moderator for today, who will start us off with an overview of retinal pathology for this session. Welcome Professor Kim Hemsley, who heads the Childhood Dementia Research Group at Flinders University in Australia. And huge thanks for being up with us at a very unpleasant time. So thanks for joining, Kim.
Thank you very much, Cara. I'm very privileged to be here, and I think it's going to be a fantastic session, as you mentioned. So getting right into it, if I could have the next slide, please. The reason we're interested in the retina and talking about it today is because it's actually part of the central nervous system. Generally, when we think of the CNS, we think of the brain and the spinal cord, but the retina is part of the CNS as well. And on the diagram there on the right, you can see that the retina is the sort of reddish coloured area at the back of the eye. And there's some detail to that I'll show you in a moment, but the retina communicates with the brain through the optic nerve, also shown in that diagram. Next slide, thanks. So both the retina and the brain deteriorate in Sanfilippo, and the treatments that we're focusing on to get brain function back in children with Sanfilippo may or may not treat that retina well. So we need to be thinking about that when we're designing those therapies. An example of a treatment that may not get to the retina in sufficient quantities might be something delivered directly into the brain, for instance. So for optimal quality of life, obviously we want all of the central nervous system to be treated. So we need to start thinking about how we assess the retina in disease in patients today, but also how we incorporate measurements of retina into clinical trials and, in the not too distant future, hopefully, into post-approval monitoring. Next slide, thanks. We're going to hear three, I think, beautiful presentations. And so what I'll do next is just give you a little bit of background on what we know about retina in Sanfilippo, just to set you up for those sessions. We're going to hear about a retinal cell model from Siti Mubarokah here in Adelaide. We're going to also hear some presentations from clinicians in both San Diego and Germany, and I'll introduce everybody at the end of this session, but they're going to tell us about their use of monitoring techniques for retina in patients with Sanfilippo. Thank you. Next slide. So the retina is that area at the back of the eye, as I mentioned, and it's actually quite a complex structure. You can see that there's a number of different cells there represented in different colours and different shapes, and they all have very important functions. Thanks, next slide. The light comes in the front of the eye, of course, and it actually moves through those cell layers all the way to the back to where the rods and cone photoreceptor cells are, and this is the outer nuclear layer. In that area, a photochemical reaction occurs, and that enables those cells to communicate with the other cells in the retina, and ultimately the retinal ganglion cells, shown in purple, send their axons, send their projections back into the brain and talk to the brain. And this is how we process visual information. Next slide, please. Now, those rods and cones are quite different, similar but different. The vast majority of the photoreceptors in the retina are the rod photoreceptors, and they're the ones that are really good in low light and provide you with night vision. The cones, on the other hand, are good for the fine detail and the colour vision. Next slide, thanks. When we study the retina, and this is what you're going to hear more about in the latter two presentations in the session, we can use a variety of techniques. We can look at the electrical activity of the eye, so how the cells are talking to each other, and this can be done using the electroretinogram, or ERG. And essentially we end up with those little traces that you can see in the right-hand diagram, and that can tell skilled people like our speakers in the session about what those individual cell types are doing in the retina. Next slide, please. Now, that's not a new technique, ERG. It's been around for a long time, and in fact there's been studies done on patients with Sanfilippo syndrome in the 60s. So this was a cohort of patients with different types of MPS, but I've put a purple box there around the 2 Sanfilippo patients in this study, one 17-year-old and one 10-year-old, both of whom had night blindness. When their eyes were studied, the electrical activity using an ERG, and this is the panel on the right, you can see that compared to the normal traces, when they looked at the dark vision in these patients, those traces are definitely not normal. So the electrical activity there is abnormal. Next slide, please. In labs like mine and others around the world, we can also study the retina using tissue pathology. So we can actually remove the eye and have a look at it with a variety of different stains. Next slide, please. And one of the first studies I'm aware of that has done this in Sanfilippo mice is work by Koer Helderman, Anne Hennig, Mark Sands and co at WashU, and they looked at the MPS IIIB mouse retina. In that panel of images, you can see that there's a thinning, particularly in what's called the ONL, or outer nuclear layer. That's where those photoreceptor cell nuclei are, and that gets thinner as those mice age. And these investigators also saw that there were changes in the ERG when they measured that in those mice. So the top 2 lines reflect the changes going on under dark conditions in the ERG. And you can see that there's a separation between the control, or wild type mice, and the MPS IIIB mice at around about 15 weeks of age in those animals. In contrast, when they looked at the light vision of those, or the ERGs under light condition in those mice, which reflects those cones, they didn't see too much difference. Next slide, please. Some of our own pathology work, we've looked at the structure of the eye using a routine H&E stain. And if you look at the ONL layer there, the dark purple layer at the bottom, you can see there's quite a thinning of the outer nuclear layer in MPS IIIA mouse eye. And we've shown using what's called a tunnel stain that this is caused by a certain type of cell death, and that can be detected quite early. Those cells are starting to die quite early in that Sanfilippo type A retina, and there's progressive loss of those photoreceptor cells with time. Next slide, please. We can also drill down and look at the different parts of those photoreceptor cells. They're quite detailed cell types, and we can see that the outer segments in the top panel there, which is where the pigments that are important for the phototransduction in the eye, the rhodopsin pigment, for instance, you can see that that is thinning in the Sanfilippo retina. But the inner segment is also affected, particularly in the cones, and this is where the mitochondria, the energy producing centres of the cell, are located. Both the inner and outer segments are affected in those photoreceptor cells. Next slide, please. But the damage doesn't seem to be occurring very much in the other cell layers, which is good news. So we sort of know where we have to target our therapies. We looked at all the other layers of the eye, list in the Sanfilippo A mice, and we couldn't see much difference there. Next slide, please. So do the changes in retina equal those in brain in Sanfilippo? Well, our view is that yes, they largely do. You can have a look at the heparan sulfate build-up in retina and brain, and it's remarkably similar. The same with the inflammatory response. So microglial activation shown there at the bottom. And next slide, please. So we map those, and yes, they're remarkably similar. So retina does seem to give us a good indicator about what's going on in brain. Next slide, please. And we can also study the retina using a visual image, and you'll hear about this in the next couple of talks. So a modern technique is called optical coherence tomography, or OCT. And many of you have had this done. If you wear glasses, you'll have had this done. It's essentially an image of your retina that can be coloured like the photo there. It can be done on infants all the way through to adults. Next slide, please. And in Sanfilippo retina, we can see a reduction in the outer nuclear layer where those photoreceptors are with an OCT, so it's exquisitely sensitive. Next slide, please. And we can also detect the effect of treatment. So in this series of graphs, you can see in the green, the Sanfilippo mice, and in the purple, the treated Sanfilippo mice. These received gene therapy. And we were able to detect the preservation of retina in these Sanfilippo mice with OCT. So it's ideal for looking at treatments that can access the retina and the brain for treating Sanfilippo. Next slide. And so in summary, the retinal changes, as far as our research goes, are largely limited to the photoreceptor cells, but they do occur quite early. We can use a variety of techniques to quantify the changes in the structure and function in the mice, so likely also in patients. And we do need to increase the use of these methods to monitor what's going on in retina and to monitor the effect of treatments, because these are non-invasive, widely available and quite low-cost methods. And we need to focus on not only brain, but also retina in Sanfilippo patients for optimal quality of life. So that concludes my part of the presentation. But just briefly to tell you about Dr. Siti Mubarokah, who'll be presenting next. Siti's a postdoctoral fellow in the Childhood Dementia Lab here in Adelaide. She's an early career researcher. She graduated from Flinders, and she's been focused on developing a cell model of the retina to look at drug testing. She'll be followed by Dr. Chris Toomey, who is Assistant Professor of Clinical Ophthalmology at the University of California, San Diego. Chris is a physician scientist and a vitreoretinal surgeon, at UCSD, and he has an active surgery clinical practice and a basic science lab as well. The final presentation will be Dr. Yevgeniya Atiskova, who is a senior physician and scientific associate at the University Medical Center Hamburg in Germany. Dr. Atiskova is a pediatric ophthalmologist, and her research efforts include characterization of retinal findings, in individuals with Sanfilippo syndrome, but also has established retinal biomarkers for Batten disease and Fabry disease. So I think we're in for a treat. Thank you very much, Siti. Over to you.
Thank you very much, Kim, for the kind introduction. As mentioned by Kim, I'm currently working on a new cell model, a photoreceptor cell model, which is created from the skin of Sanfilippo type A or MPS 3A patients, and evaluating its potential as a drug testing tool. Next slide, please. We understand that like changes occurring in Sanfilippo brain, significant retinal pathology occurs in affected patients. Therefore, assessing the disease progression in neuroretina is as important and may present as a therapeutic target in Sanfilippo syndrome. In 2020, a study by Mahato et al. Identified 5 small molecules that can induce fibroblasts to become a functional photoreceptor cells in only 10 days, or known as CIPS, chemically induced photoreceptor-like cells. The process doesn't require viral transcription factors. They tested the method in several different fibroblasts, including mouse embryonic fibroblasts and human adult dermal fibroblasts. As shown in the diagram, to convert human fibroblasts into CIPS, the 5 molecules, valproic acid, SER 99021, Repsox, forskolin, and IWR1, are added to the fibroblasts. Then, to promote and support the formation of photoreceptors, another 3 molecules, sonic hedgehog, taurine, and retinoic acid, are added from day 5. The images on the slide showed positive expression of specific photoreceptor markers such as recoverin and rhodopsin after the conversion of human fibroblasts. In their study also, CIPS culture from mouse embryonic fibroblasts can be maintained to be mature in differentiation medium containing 5 small molecules after the 10 days conversion. Next slide, please. In our lab, we adapted their protocol to convert patient fibroblasts into CIPS. Thanks to the perpetual funding, we are able to recruit Sanfilippo patients and established generation of patient fibroblast culture in our lab. They are extremely precious cell lines from classical or rapidly progressing MPS IIIA and the slow progressing or attenuated MPS IIIA and the healthy patients. The diagram shows how we established the primary culture, expansion of the culture, and characterize the disease phenotypes by enzyme activity and determination of heparan sulfate level. Next slide, please. With small modification, we applied the chemical reprogramming method to convert patient fibroblasts into photoreceptor cells. Fibroblasts were seeded at day 0 at high density and left to be post-confluent for 3 days. Then we started to induce the fibroblasts from day 4 for 10 days until day 14. We then differentiate SIPS culture in the differentiation media containing small molecules. The images show the initial fibroblast morphology and the SIPs after reprogramming, which most appeared as cells with a round soma. The experiment in our hands support the study by Mahato et al. By showing that conversion of patient fibroblasts into SIPs results in increased expression of photoreceptor-specific genes such as CRX, recoverin, and rhodopsin. We also showed significant downregulation of fibroblast-specific markers, collagen 1A1 and CTGF in SIPs. Next slide, please. Lysotracker staining is used to show enlargement of lysosomal compartment size in the disease cells. The top images show significantly increased lysotracker intensity of MPS IIIA fibroblasts compared to normal, which also shown in SIPS culture. Heparan sulfate levels on the right graphs confirms the substrate accumulation in both MPS IIIA fibroblasts and MPS IIIA SIPs. Next slide, please. We also observed upregulation of several other disease markers in Sanfilippo SIPs, such as lysosomal marker LAMP1, autophagy marker LC3B, mitochondrial marker TOM20, cell death marker caspase3, and inflammatory marker such as INOS and NF-kappa beta. Next slide, please. Currently, while many human clinical trials are underway around the world, the admission is invariably limited to those with classical rapidly progressing disease. This leaves a patient with slower but nevertheless devastating disease without hope, unfortunately. So here, to demonstrate SIP's capacity as a model for drug testing, we reprogram attenuated MPS fibroblasts into SIPs and test an FDA-approved neuroactive drug, then evaluate the treatment impact on the disease readouts. We used 5 attenuated cell lines with 1 classical and healthy control. The drug we used is still, we still label that as drug X because we are still conducting more assays to support these preliminary findings. Next slide, please. We incorporate the drug treatment during SIP's extended culture in differentiation media for 10 days for this drug X treatment. The drug treatment started after fibroblast conversion into SIPs. The differentiation media containing small molecules and test drug was applied fresh every 2 days until they're harvested. Next slide, please. Staining of the MPS IIIA SIP showed not only rhodopsin positive cells, but we also found GFAP positive cells And we view this as a positive findings as both neurons and glia are affected in MPS IIIA. mRNA analysis show upregulation of GFAP expression in SIPs, and as expected, GFAP expression is higher in MPS IIIA, both classical and attenuated, compared to the healthy control. On the top two graphs on the slide shows increased expression of recoverin and significantly down regulation of fibroblast marker CTGF in all SIPS cultures that further confirm the conversion process. Next slide, please. These graphs show the effect of drug X treatment on the disease markers of individual patient SIPs. Most patient SIPs seem to respond positively to this drug treatment, which is exciting. And this data also potentially shows therapeutic target of drug X in MPS IIIA, such as lysosomal function, but the reduction of LAMP1 expressions might also target autophagy by reducing the level of LC3B and P62, and hopefully also improve cell recovery by the reduction of cell death marker caspase-3, and improves inflammation by reducing the NF-kappa beta level. So we have further assays to determine deperon sulfate level, which is currently underway. Unfortunately, not enough time to have that data to be shown in this presentation. Next slide, please. We plan to test more drugs using this model system. Drugs include anti-inflammatory, anti-apoptotic, antioxidant, sphingolipid substrate inhibitors, growth factors, and many more. Next, please. In summary, SIPS culture is a disease-relevant photoreceptor-like cell model of MPS IIIA. SIPS culture is rapid to generate, so it's very useful tool. And it also contains raw positive cells in the culture, MAP2 positive neurons, and GFAP positive cells that might show that the glial cells also present in the culture. So it's a mix of culture. And individual patient cell lines of SIPS culture express disease markers. So that's good. That means that once we convert fibroblasts into SIPs, they're still carrying their disease characteristic, and we want that. SIPS culture is also amenable to rapid drug screening experiments in the individual patient cell lines, and it could be used to model any lysosomal storage disorder or childhood dementia with the photoreceptor pathology. Next, please. We would like to thank and acknowledge all the patients and families who participated. Dr. Nicholas Smith, who is also a co-chief investigator in this project, and Nurse Louise James from Women's and Children's Hospital. Dr. Lisa Melton from Sanfilippo Children Foundation, everybody in the Childhood Dementia Research Group here at Flinders University, and Dr. Lian Wina, former member of the CDRG team, and then Associate Professor Martin Snell and Dr. Paul Trim for their help with the heparan sulfate measurements. We also would like to thank for the support from the Sanfilippo Children Foundation, Cure Sanfilippo Foundation, Little Heroes Foundation, and the Perpetual. Thank you very much.
Oh, okay. I guess I'll start. So today I'm going to talk about whether retinal imaging can be used as a non-invasive biomarker for Sanfilippo syndrome. So to introduce myself, I'm a retina surgeon and a physician scientist here at UCSD. And so my primary clinical and basic science interest is in age-related macular degeneration. And in this disease, I've been studying heparan sulfate for the couple decades now, well, almost a decade now. And I was introduced to Sanfilippo syndrome through a longstanding collaboration with Jeff Esco, who studies heparan sulfate here at UCSD. And so one day, Jeff knocked on my door and he asked me, so do you think retinal imaging could be used as a non-invasive biomarker for Sanfilippo? And so I had some knowledge of Sanfilippo syndrome, but over the years, I've been educated by Jeff and Cara and other members of the Sanfilippo community, and I've tried to, in return, offer some of my expertise in retinal imaging and AMD clinical trials to the Sanfilippo community. And so today I'll introduce this topic and then show some of our imaging that we've done in patients. We go to the next slide. So this concept of retinal imaging was as a biomarker for neurodegeneration, as Kim kind of introduced, really originates from the fact that the retina is an extension of the central nervous system. But also optically, it's just ideal for imaging. There's been, millions of years of evolution that have, created this optically perfect eye. And so you have a optically clear cornea, you have an intrinsic lens to focus light on your retina. And so it's really ideal for the high resolution imaging. And also, what's a major highlight for the Sanfilippo community is that retinal imaging is non-invasive and it just takes about 10, 30 seconds to get an image. And once you buy the machine, it's pretty cost effective as well. So next slide. The most common imaging modalities that are used clinically in retina imaging is an optical coherence tomography. OCT is used in short, which can show some really detailed images of the retina with about 10 micron lateral resolution. So here I'm showing a macular OCT from a Heidelberg instrument. And then also wide field fundus photography is very common as well. And so here I'm showing you an autofluorescence wide field imaging from an Optos instrument. And we'll come back to some of these as we show images in some of our patients. And so those are two of the kind of more modern imaging technologies that are used. In using these imaging modalities, we've been able to, or the community has been able to show, well, they've been able to detect small changes in the retina in neurodegenerative conditions. So for example, stroke, multiple sclerosis, Alzheimer's disease, and Parkinson's. They've all been able to show some changes in the retina that correlate with the neurodegenerative conditions. It's noteworthy, though, that these changes tend to be small, they tend to be variable, and it's really limited kind of widespread adoption of these imaging modalities as a true biomarker for these neurodegenerative conditions. Next slide. It's notable, though, that in eye disease, these imaging modalities have led to FDA approvals as a primary endpoint. And I know, from listening to these talks and talking with other members of the Sanfilippo community, that FDA approval is a topic of great interest. And so here I'm showing some data from the Oaks and Derby trial, which led to the FDA approval of Syfovre for geographic atrophy and age-related macular degeneration. So what you're seeing is some fundus autofluorescence images that show progression of lesions that are highlighted in brown over time. And Syfovre was approved by the FDA for reducing the autofluorescence progression rate of these lesions by about 20% over a 2-year period. And this is important because this imaging was the primary endpoint for approval, and other clinical measures like visual acuity were not significant in this study. And so you'll see some more fundus autofluorescence images when I come back to some of our longitudinal data, or some of our data in MPS patients. And so next slide. And so I was asked by Jeff and Cara this past year if retinal imaging could be used as a biomarker. But of course, this is entirely dependent on a progressive retinal phenotype. And so although there's some documentation of retinal phenotypes in MPS, there's kind of been a lack of longitudinal data, using really modern imaging technologies. And so initially what we did was we queried our UCSD retina practice genetic database for patients with MPS mutations. And so we found a patient with a mild presentation of a multisystemic disease and compound heterozygous mutation in the heparin alpha glycosaminidase gene with about 8 years of follow-up imaging. And so I'll show you some of those images next. We can go to the next slide. And so here's a fundus autofluorescence image over 8 years. So the most striking imaging was found on fundus autofluorescence imaging, which, as I mentioned before, notably is the primary endpoint for that Syfovre FDA approval. And so here you can see the optic nerves kind of off to the side here, but I don't have a pointer, but so you could see in the mid-peripheral retina, there's this greatly hypoautofluorescent pigmentation that's happening. And then there's a parafoveal ring of hyperautofluorescence, so the white lesion that's kind of right in the center there. And then over time, as the patient progresses, you see these hypoautofluorescent lesions that are expanding at a rate about 1 to 2 millimeters a year until their last follow-up in 2023. And so those hypoautofluorescence imaging, which you can see in black, see these little dots that are in black, and as they grow over the years, those represent loss of the retinal pigmented epithelial cell layer. And functionally, that's a blind spot for the patient. And so it just so happens that the retinal pigmented epithelium has an autofluorescent pigment called lipofuscin in it. And when that cell layer dies off, you lose your autofluorescence signal. And so that's what that black lesion that's progressing over the years is. And that's the exact lesion that was highlighted in brown and tracked over the years for the FDA approval of Syfovre. And can we go to the next slide now? And so the other eye, that I showed you the right eye before, the left eye had really symmetrical findings. So you could see this early hyper-autofluorescent ring in that parafoveal region, so the white kind of circle in the middle, followed by this hypo-autofluorescent lesion growth, the black lesion that's growing at about 1 to 2 millimeters per year until their last follow-up. And so go to the next slide. And so it's noteworthy that these lesions were also visualized on optical coherence tomography, as well as the infrared photos as well. So you can see here the progression of the right and left eye on the infrared photos on the very left-hand side. And then these optical coherence tomography slices, you can see the lesion in white. You can see the lesion growing over time, but you're just getting certain cross sections through it. But you can quantify this as well, just like you could have quantified the fundus autofluorescence images. And next slide. So getting back to our initial question, can retinal imaging be used as a biomarker? It certainly looks very promising. These wide-field fundus autofluorescence images show clear progression in this particular patient over time, and it is very promising because fundus autofluorescence imaging has been used as a primary endpoint in prior geographic atrophy trials. Next slide. So in terms, though, of the next steps, we really need a longitudinal natural history study to determine the heterogeneity of the retinal phenotype that's present within patients with MPS syndromes and Sanfilippo. And as Kim mentioned before, we need to know if this is really correlating with the neurocognitive decline or not. We've begun at UCSD sort of a feasibility pilot study, and some of the early lessons that we've had is that it's very patient dependent whether you can get imaging non-invasively. So some patients, the imaging may need to be part of an exam under anesthesia. And the other issue that we've had is that MPS is, it's a very rare condition. And so there's really only a few patients that have any type of MPS in the greater San Diego area. And so that really limits our ability to recruit patients. And so off to the side here, I'm showing an adaptable imaging instrument that can be used under exams under anesthesia. It has an adaptable arm because most imaging is in the upright position. And so when you do exams under anesthesia, you need to have an adaptable arm for these imaging devices. And so last slide. And so I'd just like to acknowledge Jeffesco and Cara for their guidance and their support throughout this progress or throughout this project, and for teaching me a lot about MPS syndrome and Sanfilippo. And then I'd also like to acknowledge really how impressive it is that we have a dedicated session to eye disease in this program. And I'm more than happy to take any questions that you guys have.
Hi, good evening or good afternoon or whatever time it is at your country. I'm Yevgeniya Atiskova from Hamburg, Germany, and I'm a clinician ophthalmologist. Mostly of my clinical and research time, I spent my time with lysosomal storage diseases and mostly with NCL patients. And a few months or years ago, I started to see MPS-free patients as well. And I brought some results from a pilot study, and I was not really sure about the audience today. And seems like my presentation is a mixed repetition from the first and the third talk, but let's see, maybe there are some things that are interesting for you. So next slide. To the background, you heard most of that. Ocular manifestation in MPS-3, including their timing and severity, are rarely documented in literature. Yet a significant proportion of MPS-3 patients suffer from disabling visual impairment. And cases of optic atrophy, disc swelling, and mostly retinopathy have been reported. There's a publication from 2006, a very good review to me, and it says moderate to severe retinopathy with pigmentary changes is a prominent feature of MPS-3, and corneal opacification is not usually significant. Optic atrophy and disc swelling can occur, and an 8-year-old with MPS-3 presenting with diffuse corneal opacity and peripheral cataracts has been described. But the referred publications are about cases which were not genetically confirmed. So I included some publications about MPS-3 patients with a genetically confirmed diagnosis. And one of this is in a cohort of 12 MPS patients with an attenuated phenotype of MPS-3A. A group observed that 9 of these 12 MPS patients developed retinopathy or visual impairment, which was not quantified or imaged. So the group described that 9 out of 12 patients suffered from visual impairment or retinopathy was seen by an ophthalmologist, but there were no images or no quantification or numbers of visual acuity in this publication. Next. There's one case report from 2015 where a 29-year-old patient with retinitis pigmentosa-like changes in the visual acuity with 20 out of 120 out of 80 was detected. And as you see on the retinal images, a breadcrumb-like pigmentary retinopathy with perifoveal pigmentary involvement was shown. Next. As you see, this is an OCT of this patient, and the OCT showed a loss of normal neuroretinal lamination, degeneration of the outer retinal layers, and also cystic macular changes. And the electroretinography, which is not shown here, exhibited a generalized retinal rod cone dysfunction. Next. Another case report with a genetically confirmed MPS IIIA case, a 53-year-old male patient showed also breadcrumb retinopathy. The middle picture is autofluorescence, where you also can see some changes in the central retina, and the right picture is an OCT, which shows a massive retinal degeneration, and you can't really see the single layers, but the outer retinal layers are totally destroyed on this image. Next. Here are some information about some mouse models. I go through that quite fast, as we heard in the first talk, a lot of this. But this is one of a publication I liked a lot, and they did, maybe that's also a good example to distinguish OCT from ERG. This was one of the questions. ERG is a functional measurement, and OCT is a morphological measurement, and that is a big difference. As EMA, FDA are always asking for functional measurements, but morphological measurements are also very, very important, as they can distinguish even slight differences between progression. And on these images, you see immunohistochemical investigation on the left side, which is similar to an OCT. An immunohistochemical investigation is way more detailed than an OCT, as you can image and stain the single cells and not only the layers. And on the right side, you see a photopic and a scotopic ERG, which means that in dark adapted and light adapted situations, the retina function was measured. And in this particular publication, you see that the 9-month-old MPS IIIA mouse has reduced rods, reduced cones, reduced outer nuclear layer, reduced inner nuclear layer, and also in the functional measurements, the A wave in the scotopic ERG is reduced, which means that the rod photoreceptor function is reduced, and also the B wave is reduced, which means that bipolar cell function in rod bipolar cells is reduced. And also in the photopic ERG, you see that the B wave is significantly reduced, which is a measurement for bipolar cell function in the cone of bipolar cells. The A wave of the cones is not that easy to compare in a mouse model, as the amplitudes are very, very low, and the differences are only small. So in my opinion, we have done a lot of ERGs in NCL mice and different NCL types, and the A wave in the photopic ERG with the which measures the cone function, and cones are only 3% of the photoreceptors in mice, is not that you can't really good compare it in the wild type with the MPS III mouse. Next. Another publication focused on the MPS IIIB mice and demonstrated also progressive rod cone degeneration. We heard about that from Kim Hemsley, more detailed, so I would just go to the next slide. And here you can see another publication which came out last year, and it studies MPS IIIC mice and found significant retinal thinning, photoreceptor cell loss, and a disorganization of retinal layers. You see that cones were imaged and rods were imaged, and both were reduced compared to the wild type mouse, and also the outer nuclear layer thickness and the rows of the photoreceptor nuclei were significantly lower in the MPS IIIC mouse compared with the wild type. Next. This group also had a histology of a patient, and you see in the control a healthy patient and the MPS3C patient in the lower picture, and you see a reduced outer nuclear layer and also inner nuclear layer. And the inner nuclear layer contains mostly bipolar cells and some other cells like amacrine cells, horizontal cells, and both layers are reduced in this post-mortem analysis of an MPS3C patient. Next. I think we can skip that. It was in detail explained by Kim Hemsley. Yeah, and what was the aim we did in the last weeks and months? As the detailed retinal phenotype, the incidence and time course of ocular involvement and visual deterioration in MPS3 is still unknown due to cognitive impairment and therefore lacking compliance for visual testing and ocular examination. And it is a heterogeneous disease and a very rare disease. It is quite difficult to collect data on this. Our aim was to undertake a comprehensive and systematic analysis of the retinal phenotype in a cohort of individuals with MPS3, utilizing SD-OCT imaging, mostly in sedation, as our patients most of our patients we see in our hospital are children, and most of them have a severe neurocognitive phenotype, and they are not able to perform an upright investigation. All the sedations were necessary for other investigations, for example, for MRI examinations or for small operations of ears or teeth or anything, and we used the sedation to collect data on the retina. And these findings may provide a novel insight into the complex disease mechanism of retinal degeneration in MPS3 patients and support future diagnostic and therapeutic strategies. Next. We tried to measure visual acuity, and as I was working a lot with NCL patients, there are some different methods you can apply in neurocognitive impaired patients. In the patients we investigated so far, we tried to do visual acuity tests as it is displayed above, the usual visual acuity test, but this is very challenging, especially in severe neurocognitive phenotypes. It's impossible. So another thing that we are starting right now, I don't have data on this which I can present to you right now, but we did this in CLN2 patients who are also cognitive impaired, and the children can sit on the laps of their parents. And you have two different charts, gray part and a stripe part, and the natural reaction of a child or a patient who is cognitive impaired and who sees this striped and this gray field is to preferentially look at the striped field. And then the stripes are getting smaller and smaller, and as soon as the patient can't distinguish the stripes anymore, both fields seem identical to him, and he do not have a preferential looking anymore. So if there is some concentration and they have compliance to do this test, it's way easier than reading letters or numbers or naming pictures. And we try to establish this in MPS3 patients right now. Next. We also try biomicroscopy of the anterior eye segment, either in an upright position or sedation to look at the corneal opacification. Next. And we also try to do fundoscopy investigations, which is also almost impossible in the patients we see, as the compliance is quite low. Next. Another investigation which Professor Toomey presented to you is the OCT. You can perform it in an upright position. On the left picture bar, the patient sits in front of it, or you can do that in a lying position under sedation. And we have this gadget. We investigate a lot of NCL patients with that. And we also try to investigate all the MPS-free patients who are under sedation because of different reasons. And please go back. Please go back. In the lower picture, you see that we measure a retinal nerve fiber layer. That's the nerve fiber layer around the optic nerve head, and the retinal thickness. Next. And we have investigated 16 patients so far and analyzed this data, and 6 of the patients were investigated, but data were not analyzed so far. And the inclusion criteria for us were diagnosis was genetically confirmed of MPS3, and the OCT was performed. So not all these patients we included were able to do visual acuity testing, but we tried to include the preferential looking test by now, and the neurocognitive phenotypes were clinically graded in attenuated and severe. Next. The visual acuity in the patients we could investigate quite good was not reduced, yet only attenuated patients without severe cognitive impairment could be assessed by the usual visual test. There was no obvious corneal opacification in all these patients we investigated so far, and also all the patients had a normal nerve fiber layer. All the patients with a severe phenotype showed retinopathy, for example, central retina thinning, parafoveal atrophy of the outer retinal layers, foveal ALM thickening, or a combination of these. And 3 out of 5 patients with attenuated phenotype had a parafoveal atrophy or outer retinal of the outer retinal layers or foveal ELM thickening. I brought some patients' examples. Here you can see MPS3A patient, 3 years old, with a severe neurocognitive phenotype and a massive central and peripheral retinal atrophy with a massive degeneration of the outer retinal layers. Next. And here you can see an MPS3B patient, 8 years old, with an attenuated neurocognitive phenotype, parafoveal atrophy of the outer retinal layers, which you can see in the at the blue arrows. So the central retina looks quite normal, but parafoveally you see a massive degeneration of the outer nuclear layer. Next. Here's an example of a MPS3B patient with an attenuated neurocognitive phenotype, which who had a visual acuity 20 out of 25, which is quite good. And the arrow shows the foveal ELM thickening. Usually this is only a small line. And this is a thickening we could see in a lot of patients. Next. And the last patient example I brought is an MPS3A patient with a severe neurocognitive phenotype and a parafoveal atrophy of outer retinal layers. Next. So the perspective of our study and of the things we do right now, which are, still under development, is that parafoveal atrophy of outer retinal layers could be shown in 63% of MPS3 patients, foveal ELM thickening in 69% of the MPS3 patients we investigated. And all the patients with a severe phenotype showed a retinopathy. And longitudinal investigations and large cohorts are necessary to evaluate the onset and the progression, and also maybe some similarities between patients. And as clinical trials are upcoming, a systematic ophthalmological investigation and natural history collection regarding retinopathy is important to address blindness in MPS3, what Professor Toomey already said. So yeah, thank you for listening. And yeah, I read some questions by presenting. Were coming up.
Thank you very much to all of the speakers in that session, Siti, Chris, and Yevgeniya. I think what we have come away with is that we don't quite have enough information about what's happening in the retina in patients with Sanfilippo. And so there are some challenges there that it would be great to discuss further. Moving through some of the questions that have been asked. A question is about an opinion on what is best to use to capture outer nuclear layer changes. Is that OCT, ERG, or something else? And maybe if I throw to you, Yevgeniya, and
yeah, maybe I also tried. I already tried to answer that. It is, to me, there are two totally different investigations. One is a functional measurement, and the other one is morphological. So you need both. For mice, I always would go with immunohistochemistry, which is similar to OCT but more detailed. But in patients, we can't do that. And so the OCT shows us which retinal layers can degenerate and in which time. And the ERG is a functional measurement. And yeah, maybe you can ask whether a visual acuity measurement or ERG measurement would be a better functional readout. And also this question is quite difficult, as visual acuity only catches the central retinal vision, and ERG is catching the function of the single retinal cells. Also, this is a combination of a visual field testing, a visual acuity test in ERG, and an OCT measurement would be best for me. But if the patients weren't cognitive impaired, I would do all of these. But as our patients, we don't have 53-year-old patients where you can do an autofluorescence in an upright position. I never seen that in our cohort. And as we have mostly cognitive impaired patients, you have to try to find at least one functional measurement and one morphological measurement. And for me, it is the visual acuity and the OCT. As ERG in an upright position, it is not really possible in our cohort. And ERG under sedation is very variable, as the sedation has an impact on the ERG. And visual field testing is not doable in our cohort at all. I don't have maybe 2 patients out of this 20 which could maybe do a visual field testing.
Thank you, Yevgeniya. What about your views, Chris?
Yeah, to me, it's where the pathology is. So ERGs take like the whole retina, basically. And for the most part, a full field ERG is looking at peripheral retinal changes. And so if those are there and they're changing a lot, you're going to pick that up on an ERG. But if, for the most part, a optical coherence tomography is focused on the macula. And if there's changes in the macula, you're going to pick those up way more easily with an optical coherence tomography than you are with a full field ERG. If you did a multifocal ERG, which would be impossible in these patients because of fixation and everything, you potentially could pick up macular changes, but that's out of the question really in this patient population. So yeah, I mean, what's the correct thing to do? If you had a Heidelberg instrument with an arm that could get under exams under sedation, that could get optical coherence tomography and autofluorescence imaging, I think those are basically what you can do in a patient population like this. We did try to do this in non-invasively but it's very patient specific, and so that's really what we learned in a small kind of pilot study, was that not all patients can, or actually most patients can't, focus for an imaging clinic. So.
Thank you, Chris. Another question was why there isn't, I guess, a better data set for Sanfilippo patients, a better understanding about what's happening in the retina, and how might we collect that data, harmonise that data. Do either of you have ideas about how we can do that as a Sanfilippo community?
Yeah, there are lots of great examples of this. So I think one of the best is I'm a part of the macular telangiectasia project, and this is also a pretty rare disease, but there are numerous sites internationally. They've collected about 3,000 patients and have long-term history data over the years. You're sharing images, and so that involves hiring clinical staff to do these things. So it does require requires money. So that's kind of one of the barriers. Yeah, and so anytime you're going to be sharing cross-institutional data, you have to, at least at our institute, you have to have RBs and other things like that, and that requires the staff to be involved, and so it kind of changes in my mind. Once you start to move to multi-institutional things, it just requires more resources.
One thing I've noticed just very practically is, and so I'm speaking, U.S. based, most patients don't necessarily live near a major research medical center. And even if they do live near a children's hospital, the likelihood that they can easily, or their team easily coordinates with an ophthalmologist that is able to come when they are having opportunistic sedation events. These are kind of the logistical challenges that I think we run into with collecting that data. I'm not sure I have the answer, but I don't know if you guys have any, your thoughts on how to overcome some of those kind of issues.
Yeah, I know, Cara, we've talked about this a lot. And so, the idea is you create hubs. So, you have a hub in Duke, you have a hub at, UCSD in San Diego, you have hubs, at places that have a lot of patients. And so you start off with a small number of institutes that have, more higher volume. And then you kind of move into, other places as you can. Yeah.
Yeah, the Hamburg site really, they have a very integrated system. I'm always really impressed by what you guys are able to accomplish in Hamburg. So, kudos to the team there.
Yeah, it sounds not bad, but if you knew how it works, it's like you get a phone call, then you have to think, I have to run to the children's hospital as the patient is under sedation there. Then they have to apply eye drops so the patient get a white pupil. That's in most cases somebody forgot that. Then you have to have this OCT gadget in the pediatrics department. It's mobile, but it's not mobile. So when you don't have a mobile OCT gadget which can measure the patient in the lying position, and I think we're in Hamburg, we are one of 5 clinics or maybe 10 clinics who even has this machine, and all the other clinics have this machine in the ophthalmology, and no MPS III patient is sedated in the ophthalmology, because why? So we can only do that because we had a study on CLN2 patients, and we moved this gadget to the pediatrics department. And I am an ophthalmologist who knows what should be done, or I try to find out. I'm still in the learning process. Which investigation should be done, and how you can do that in the MPS III patients. And it just came to my mind after investigating 20 patients that we should stick to preferential looking. I don't know if it works, but I would try that out. So I think you can't compare it with McTell, for example, because McTell, all the patients can do examination at any ophthalmologist who has an OCT and maybe an autofluorescence, and these patients, or at least our patients, they can't do anything without sedation, most of them. And then you have to be an ophthalmologist who is at the right time on the right spot and has this machine, which is also not that easy. So for me, I thought about recruiting somebody in Germany, and I didn't find anybody who is willing, or for who it is possible to investigate MPS III patients in the systematically manner.
So we have some more hurdles to accomplish, but I think the first step is talking about them together and figuring out, like, trying to problem solve. But CT's exploring with an attenuated phenotype, and then we hear about adult patients, and so maybe those are good places to start, wherein we don't necessarily have the same other measures to fall back on, which are being explored now in the classic severe, which is the cognitive phenotype. But maybe we can utilize this in the attenuated phenotypes where maybe sedation isn't required up front, and a good starting place.
Yeah, I always feel like if you just start somewhere, you kind of have to walk before you run. And so if you just start by, the easy things like Cara talked about, that's kind of what we did. We started with more attenuated phenotypes, people who can handle, in-office imaging. And then you try to move to, exams under anesthesia and other things as you can.
And it also is, as we have a lot of severe cognitive phenotypes, it even didn't come to my mind to do autofluorescence, as the first patients we investigate were in the eye hospital, and they even couldn't fix this point of the OCT machine. So it even didn't come to my mind to try autofluorescence. And after some investigations under sedation and maybe some attenuated phenotypes, then I thought, that would be very useful to include autofluorescence as well.
Yeah,
For me, it's still a learning process. I think we have a quite good protocol right now, but developed over a year.
Yevgeniya, you've been doing some work in Batten's disease. Are they using retinal imaging or ocular biomarkers in the Batten disease field? Are you aware?
We published also some studies where we found out that the retinal phenotype and also the retinal nerve fiber layer is correlating significantly with the cognitive and the motor skills. So there is in the CLN2 field, in the CLN3 field, there is a positive correlation that the neurodegeneration in the brain is progressing in a similar manner as the retinal degeneration. And there are also some studies, but I'm not that familiar with them, that show that Parkinson's disease, Alzheimer's disease, and also multiple sclerosis has a correlation with the OCT biomarker. retinal thickness and retinal nerve fiber layer. So retinal degeneration and brain degeneration seem to be together in several neurological diseases. And as we could show that the severe phenotypes all have a retinopathy and the attenuated phenotypes not, it could be a hint that it could be maybe, and also your mouse model says that it could be, that there could be a correlation between brain and retinal atrophy.
Okay, I think we might be running a couple of minutes behind, so maybe it's time to wrap this session up. I'd like to thank all of the speakers in the session, Siti, Chris and Yevgeniya. Fantastic presentations, and hopefully we can continue the discussion in the Sanfilippo community going forward with the hope of exploring the changes in the retina in the patient population in some more detail, and hopefully incorporating retina as a biomarker into clinical trials and post-treatment monitoring in the future. Thanks very much for your time and great presentation.
Thanks, everyone.
Thank you.